Hello friends, welcome to PVIC Breakdowns. In our previous video, I introduced what amino acids are and described their general structure. In this video, I will be discussing the classification of amino acids. Now, the 20 common amino acids can be classified based on their 1. Nutritional importance. In this system of classification, the amino acids are classified based on the ability of the human body to synthesize them. So amino acids that are synthesized by the human body are called non-essential amino acids, and they include glycine, alanine, serine, cysteine, tyrosine, asparagine, glutamine, arginine, aspartate, glutamate, and proline. You can see here I'm just using the 3-letter abbreviation of the amino acids. While amino acids that cannot be synthesized by the body and so must be obtained from our diet are called essential amino acids, and they include valine, lysine, tryptophan, phenylalanine, methionine, threonine, histidine, leucine, and isoleucine. Also, 6 of the non-essential amino acids are considered conditionally essential, and they include arginine, cysteine, glutamine, glycine, proline, and tyrosine. What this means is that normally these 6 amino acids can be synthesized by the body, but in a condition of severe illness, stress, or injury, the body might not be able to produce these amino acids in sufficient amounts. And so, to compensate for this inadequacy, they must be obtained from diet or nutritional supplements. Hence, the term conditionally essential. And note that the term essential and non-essential does not mean that essential amino acids are more important than non-essential amino acids. They are all important. It's just that our body can produce these ones but cannot produce these ones. It's just like when you have something, you're not that bothered. But when you don't have that thing, getting it now becomes essential to you. 2. We have classification of the 20 amino acids based on the chemical and structural features of their Aru groups. These red portions are the Aru groups of the amino acids. 3. We have classification based on their biosynthetic pathways. And 4. Classification based on their catabolism. Now, of all these systems of classification, the one that is of more interest to us and I will be discussing in details is classification based on the chemical and structural features of their Aru groups. In our previous video on introduction to proteins and amino acids, I said that the Aru group or side chain of each amino acid is different. However, the Aru groups of some amino acids are similar in terms of charge, structure, and polarity, and so can be grouped together. Before we move further, let me quickly explain to you what polarity is. In chemistry, polarity results from the unequal distribution of electrons in a molecule. For example, this is hydrogen fluoride. You know, this is a covalent bond, and a single covalent bond represents a pair of shared electrons, something like this. This is the pair of shared electrons. So, because fluorine is more electronegative than hydrogen, that is, it has more tendency to attract this shared pair of electrons to itself. There is now unequal distribution of electrons between both atoms. And as a result of this, the fluorine atom acquires a partial negative charge, while hydrogen in turn acquires a partial positive charge. These partial charges are represented by this symbol. This is delta minus and this is delta plus. It is a partial charge because there is no complete loss of electrons by hydrogen nor a complete gain by fluorine. It's just that the electrons are pulled closer to the fluorine atom than they are to hydrogen. However, for example, in this molecule, say we have this, or let's say here, there is equal distribution of electrons between the atoms because there is none or very small electronegativity difference between them. As you can see here, these are similar atoms bonded together. So there is no electronegativity difference between them and so no charge on them as well. For this, although they are different atoms, the electronegativity difference between them is very small and so there is no charge. So this is a polar covalent bond. So this is a polar covalent bond, while this and this are non-polar covalent bonds as there is no polarity. And one thing you should know is that polar molecules are usually soluble in water, while non-polar molecules are not. And this is because polar molecules are able to interact with water through a type of interaction called hydrogen bonding. And hydrogen bonding is an interaction involving a hydrogen atom located between a pair of more electronegative atoms such as oxygen, nitrogen, or fluorine. For example, for example, this polar molecule can form hydrogen bonds with water. This is the hydrogen bond represented by these dotted lines. So as you can see here, because of the large electronegativity difference between the nitrogen atom and the hydrogen atom, the nitrogen atom then acquires a partial negative charge and the hydrogen atom acquires a partial positive charge. The same thing applies here. So due to the partial positive charge on the hydrogen atom, it is attracted to atoms that carry a negative charge, as you can see here as well. So this polar molecule is soluble in water because of this hydrogen bonding interaction. But non-polar molecules are unable to form such interactions with water and so are insoluble in water. For example, in this non-polar molecule, hydrogen bonding with water will not work because the electronegativity difference between carbon and hydrogen is very small and so there is no charge on them. Hence, the hydrogen atom cannot attract the oxygen atom of this water molecule and therefore no hydrogen bond is formed. So the polarity of the arrow groups of these amino acids varies widely from nonpolar and hydrophobic, that is water insoluble, to highly polar and hydrophilic, that is water soluble. So based on the chemical and structural features of the arrow groups amino acids can be grouped into 5 main classes. The first class we have here is the nonpolar aliphatic aryl groups. The aryl groups of amino acids in this class are nonpolar and hydrophobic. Evidently, from the explanation I just gave on polarity, you can see why the aryl groups here are nonpolar and hydrophobic. The aryl groups here consist mainly of CH bonds, that is carbon hydrogen bonds, in which there is very small electronegativity difference and so no polarity. And since there is no polarity, formation of hydrogen bonds with water is impossible. Hence, the aryl groups are hydrophobic, that is, they hate water. Also, the term aliphatic means that the aryl groups have an open chain structure and not a ring structure as seen in aromatic aryl groups. So these are the amino acids that belong to this class. We have glycine, proline, leucine, methionine, isoleucine, valine, and alanine. Glycine Glycine has the simplest structure of all amino acids with a hydrogen atom as its side chain. I call proline a special amino acid because it is the only amino acid that has its amino group linked to its aryl group, thereby forming a ring structure. You can see in other amino acids, the amino group is separate from the aryl group. The sulfur carbon bond in the aryl group of methionine is nonpolar because sulfur has almost the same electronegativity as carbon, and so this makes methionine also nonpolar. Now let's move to the next class, which is the aromatic aryl groups. The aryl groups of amino acids in this class are to a certain extent nonpolar and hydrophobic because, as you can see here, their aromatic rings consist mainly of CH bonds, which is nonpolar. However, the hydroxyl group of tyrosine and the nitrogen atom in the tryptophan ring can form hydrogen bonds with water. So you have something like this. This and this. So you can see the hydroxyl group of tyrosine forming hydrogen bond with water and the nitrogen atom in the tryptophan ring forming hydrogen bond with water as well. Hence, tyrosine and tryptophan are largely more polar and less hydrophobic than phenylalanine. Now, the next class we have here is the polar uncharged aryl groups. The aryl groups of amino acids in this class are polar, that is, they are more soluble or hydrophilic than those of the non-polar amino acids. And this is because They contain functional groups that form hydrogen bonds with water. Amino acids in this class include asparagine, glutamine, serine, threonine, and cysteine. And as you can see here, the hydroxyl group in serine and threonine makes them polar, while the amide group this in asparagine and glutamine makes these two polar as well. So these functional groups, that is the hydroxyl group and amide groups, they form hydrogen bonds with water, hence making them soluble in water. Cysteine is quite different because the polarity contributed by its sulfhydryl group is relatively small. That is, the polarity is not as strong as that of the hydroxyl or the amide groups. So cysteine is the least polar in this class, and it forms weak hydrogen bonds with water. Also, the aryl groups of amino acids in this class are said to be uncharged or neutral because they do not ionize to become positively or negatively charged, as we will see in the next classes. The next class we have here is the positively charged or basic aryl groups. Firstly, the aryl groups of this class and this class are the most hydrophilic amongst the 5 classes. Now, the aryl groups of amino acids in this class are positively charged, as you can see here. Amino acids in this class include lysine, arginine, and histidine. And these amino acids are said to be basic because if you take a close look at their side chains, for example here in lysine, you will see a nitrogen atom which acts as a base by accepting a proton. This is what I mean. Let's say we have this. The nitrogen here has a pair of lone electrons which it can donate to a proton, also called hydrogen cation, in order to bond to it. So you have something like this. So the nitrogen atom is acting as a base by accepting this proton. And since nitrogen is the one donating its lone electrons for this bond, it bears a positive charge, as you can see here as well, and here too. The last class we have is the negatively charged or acidic aryl groups. The aryl groups of amino acids in this class are negatively charged, as you can see here, and they are also polar and hydrophilic. Amino acids in this class include aspartate and glutamate, and these amino acids are said to be acidic because if you take a close look at their side chain, you will see that they have a carboxyl group. Let me make it elaborate. So you have something like this. And a carboxyl group behaves like an acid because it can gladly donate or lose its hydrogen atom. So this is what gives these amino acids their acidic properties. And this is the ionized form of aspartate and glutamate. In the unionized form, that is when the carboxyl group still has its hydrogen atom, this is called aspartic acid and this glutamic acid. But in the ionized form, this is referred to as aspartate and glutamate. Finally, in addition to these 20 common amino acids, there are so many less common ones. Some of them are modified amino acid residues. That is amino acids that are derived from the common amino acids during prosynthetic modifications. Prosynthetic modifications. Examples of such amino acids include 4-hydroxyproline, which is a derivative of proline, 5-hydroxylysine, which is a derivative of lysine, and so many others. Now, let me quickly explain to you what prosynthetic modifications are. Now, when a new protein is synthesized, it is folded and processed into its biologically active form. However, some proteins cannot attain their biologically active form until they have been modified via one or more processing reactions. We call these processing reactions postsynthetic modifications, also called post-translational modifications. There are many kinds of postsynthetic modifications. For example, in some proteins, short carbohydrate chains are attached to some of their asparagine, serine, or threonine residues, thereby forming glycoproteins. Now, back from explaining postsynthetic modifications, there are also amino acids that are present in living organisms, but not as constituents of proteins. These amino acids have various other functions. Examples of such, amino acids are ornithine and citrulline. These amino acids are key intermediates in the biosynthesis of arginine. Lest I forget, these are the chemical structures of the 20 common amino acids at biological pH. That is the pH in our body cells, which is about 7.0. If you enjoyed this lesson, kindly click the like button, comment, share, and subscribe to my YouTube channel for more videos.